Metabolic pathway diagrams, as well as metabolism in general, can seem really intimidating and scary, but it's actually really logical. And so, if you go in with a strategy, you can figure out a lot without having to do any sort of memorization, just by interpreting diagrams themselves. And so, here's just a little overview review of how to approach metabolic pathways, what sorts of things to look for and keep track of, and this sort of thing. So let's go through. The first thing is to determine kind of like what's your goal? What are you trying to get out of it? And so often what you're trying to do is you're trying to say, how do I get from one molecule to another molecule? And we call these molecules all intermediates because they can be kind of the start of one pathway and the end of another pathway and the middle of a different pathway because the pathways are all interconnected. And so each of these molecules we can refer to as an intermediate. Sometimes they're called like metabolites. And so these are going to be the chemical compounds that you're interconverting between. You want to find your start, find your stop, and note that your stop might look like it's in the middle of a pathway or the start of another pathway. And then you want to go step by step through those different arrows and look for important things. Sometimes it can help if you're trying to focus on a specific set of reactions. Maybe circle those reactions, highlight those reactions. If you've got a digital copy, you can just kind of annotate the digital version, but don't get rid of all the other stuff because that's going to be really important too. And so we'll talk about some strategies to use when you're looking at individual reactions, as well as when you're looking at sets of reactions, because it's really important to keep that context in mind. And now let's get down to how we can actually go through and analyze them. So let's start by if we were to look at a single reaction at a time, what sorts of things do we want to look for? The first is reversibility. So is there a one-directional arrow per enzyme, or is there a kind of two arrows going different directions or a bi-headed arrow? And so often what's going to happen is that we have these double arrows are indicating that a reaction is metabolically reversible. So just by changing the concentrations of the reactant and the product, or this intermediate, you can dictate which direction the reaction is going to go. And so any place that you see that there's kind of like these double way arrows, there's one arrow going one direction, the other going the other direction, but they have a single enzyme listed with them, or you have an arrow with heads on both sides, that's indicating that a reaction is metabolically reversible. You can easily go back and forth either way. So you can think of these intermediates where you've got these double arrows with this single enzyme. You can think of these intermediates as kind of all being in a shared pool that they can convert between, just depending on the concentrations of each of these. Conversely, if you have something where you have a single arrow per enzyme, a single direction, this is going to be that you have a metabolically irreversible step. And so you can see that if you want to go the reverse direction, you actually have to use a different enzyme that catalyzes different reaction. Sometimes you might even need to do two reactions. And this is typically because the reaction is going to be really thermodynamically favorable in one direction, which means that you can't simply reverse it by changing the concentrations. You'd have to really skew the concentrations and then it would mess up everything else in the cell. So instead, you have to take an alternative reaction or reactions that are then going to make the alternative pathway overall thermodynamically favorable as well. So both of these things can happen. But as we'll talk about, there's regulations that they don't happen at the same time and kind of negate one another. But anyway, we want to look for these reversible reactions and think about the reactants that the intermediates that are shared by reversible arrows can be easily interconverted. You kind of have the pool of them. Whereas those that are separated by these one-way arrows, these arrows where there's one arrow per enzyme. So even though there's two arrows here, note that there's different enzymes for each of these, and they're actually catalyzing slightly different reactions. This one's transferring a phosphate group from an ATP onto here. This one is taking off a phosphate group, but not putting it back on ADP. It's just using water. And that way, both of these reactions are thermodynamically favorable. But these reactants are not easily interconvertible. You can't just go back and forth easily between these. You have to actually use different enzymes for both of them. And therefore, these are going to be highly regulated steps, as we'll get into later. So these steps that are irreversible are often going to be the steps that are going to be highly regulated. As we'll discuss the highly regulated steps, you want to look for the ones that have things in green or things in red, things with plus signs, things with X's. These are all indicators that you have activators. So if you have green, you have arrows with the green. You have triangles, things like this, that's going to be activating. If you have red, you've got X's, you've got the kind of like T bars, those are going to be indicating inhibition, those sorts of things. Sometimes the arrows might look like they're indicating an irreversible reaction because there is a single arrow. However, if could just be that the writers don't know, that they're being lazy, that sort of thing. So if there's a lot of double reversible arrows shown, and then one single arrow, that single arrow is probably important. But if they're all kind of single arrows, that may or may not indicate that they're all irreversible. So typically a lot of these reactions in metabolism are going to be easily reversible. And therefore we need to think about where are the regulatory points. And this also makes it more manageable to think about the regulatory points. So that is that. We have the enzyme name that's going to be written above or below the arrow or to the side of the arrow. And then you're going to have the reactants and products, which we can also refer to as intermediates, on either side. And then sometimes you have things that are kind of coming and going from the arrows. And so you have cofactors, you have co-substrates, you have things like ATP, ADP, NADH, NAD+. These can be shown based on you have kind of like curved arrows that are coming in, touching the main arrow and going out. So in this case, the ATP is coming in, the ADP is going out. Sometimes you have transient intermediates that don't actually leave the enzyme active site. And so you can see that the same enzyme is catalyzing this reaction through this intermediate that's not actually leaving the enzyme's active site. And we show that in brackets. An example is in the citric acid cycle. So we can see that we kind of have this citroyl-CoA where we stick the acetyl-CoA onto oxaloacetate before we get rid of the CoA part. And so we have this intermediate, but it's not actually leaving the citrate synthase enzyme's active site. And so we put the citroyl-CoA in these brackets. Similarly, with aconitase, we have this aconitase intermediate that we can show in these brackets. And so these are kind of helpful for helping you see how the reaction is actually progressing, but as opposed to just showing you kind of the before and the after. And it makes more sense when you actually are seeing it with the structures. And so sometimes you'll see the structures, sometimes you won't see the structures and pathways. If you do have the structures shown, what you want to do is you want to look between the different steps and think about what's changing. Are we going, say, from having an alcohol to having a carbonyl group? In this case, so malate to oxaloacetate, we go from a hydroxyl group to a carbonyl group. And so what we're doing is we're doing an oxidation, and we can see that we're using NAD+ and generating NADH. Speaking of which, you want to look for those places where you are generating the NADH, where you are generating the FADH2, where you're generating ATP or you're generating GTP. So we can refer collectively to ATP, GTP, TTP, CTP, UTP, all those nucleotide triphosphates as NTPs. And they're actually easily interconvertible in terms of kind of transferring that third phosphate group. And so you can think of them all energetically equivalent. So GTP, think of it the same as ATP, and in some isoforms, you actually do make ATP from the citric acid cycle rather than the GTP. But you want to keep track of where you have those kind of energy payouts and where you have these reducing equivalent payouts. So you want to look for when you're actually kind of spending the ATP or other NTP, as well as when you're generating it. And when you're generating or where you're using the NADH, the FADH2, the NADPH. And so that's why keeping track of them in a spreadsheet is really helpful. And so where are you generating what, and what are these enzymatic reactions? Note too, that when you have those reactions that are involving the generation of NADH, those are going to be, or FADH2, those are going to be your oxidation reactions. And if you're using them, or you're using NADPH, those are going to be reduction reactions. And those are often going to be catabolic reactions. You also want to think about where reactions are happening. And so are they only happening in certain tissues? Are they only happening in certain parts of the cell? So different tissues have different metabolic roles. And so say your liver and your muscles, they both store glycogen. So they store glucose, they hook it up into glycogen for storage. But your liver can actually break down that glycogen, so glycogenolysis, and let it out. Whereas your muscles can only break it down to this glucose 6 phosphate. They can't break it down all the way to glucose. They can't remove that 6 phosphate group. And so then they have to use the glucose 6 phosphate themselves. So they can store it for themselves, but they can't actually ship it out. Whereas your liver can ship it out. So your liver has a really important role in helping deliver glucose to the body and regulate blood sugar levels. Your liver can also make glucose from scratch through gluconeogenesis and let that glucose out. So if you are given information about the tissue, you want to make sure that you note that information and because that information could be really important. It could also be important where in the cell things are happening. And so are they happening in the general cytoplasm? Are they happening in the mitochondria? If they're happening in the mitochondria, are they membrane embedded? Are they in the matrix? And so thinking about the locations can be really important when you're looking at a diagram. For example, if we're thinking about gluconeogenesis and we're thinking about where our carbon source is coming from, if we have lactate coming into the cell, to convert that lactate to pyruvate, we do that in the cytoplasm and we generate NADH. Note that we need NADH in order to actually do gluconeogenesis. We need it for this glyceraldehyde 3-phosphate dehydrogenase step. Remember that the reason why we needed to regenerate NAD plus through like fermentation and stuff if we don't have oxygen to keep glycolysis going is because we need NAD plus for the glycolysis. So we need NADH for gluconeogenesis. If we start from lactate, we already get the NADH out here. So it makes sense then to use the pathway that's not going to generate another NADH out here. And so we would go, if we went from lactate, we could go this route and we can actually go and convert the pyruvate all the way to PEP in the mitochondria. But if we're starting from something like alanine, here what we can do is we can actually ship out malate, convert that malate to oxaloacetate in the cytoplasm, generating NADH that way. And now we have the NADH that we can use for gluconeogenesis. But if we had gone that pathway with the lactate, well, now we would have generated 2 NADH in the cytoplasm and got things out of whack. And so location can really matter. And now regulation. So some of the other key information that you can get from one of these pathway charts is the regulators. And so again, for something activation, there's different ways that you might see this. You see arrows, you see triangles, plus signs, green. Inhibition, you might see things in red. You might see kind of this T on its side, an X, kind of like these railroady looking symbols. All these things are indicating inhibition. And there are different kinds of activation and different kinds of inhibition. And they're often going to be acting on those metabolically irreversible steps. So let's talk about some different types of activation and some different types of inhibition in order to keep an eye on. So first you want to locate them. And so again, you want to look for those places where you see things in red, things in green, things with these symbols that are listed. And so these are often going to be kind of at the entry points into pathways, at branch points. So you can see we've got kind of like branch points at which we have these high levels of regulation. They can be at the ends of pathways, kind of helping drive the pathway forward. And so those are places where you want to look and see if you find those kind of key regulatory points. That's often where they're going to be. For example, this PFK1 step of glycolysis, you can see that before this step, we have a lot of different branches that we can go from. But once we do this PFK1 step, well, now we're kind of committed to glycolysis. And so this step is a branch point, and it's going to be highly regulated. Pyruvate kinase, here we have the end of the pathway, so this one's going to be highly regulated. It's kind of helping drive the reaction forward. We've got this big bang. It's going to be this metabolically irreversible step that we can't easily go back from, and so we're going to regulate it really highly as well. So how do we regulate these pathways? There are a variety of different ways, and so some of them involve allosteric regulation, and some of them involve covalent regulation. And then you also have regulation just by the levels of the intermediate, so thermodynamic regulation for those metabolically easily reversible steps. But we refer to when we're kind of regulating at the enzyme level, we can refer to it as kinetic regulation. And it can often involve either allosteric regulation or covalent regulation. And so allosteric regulation is when you have a molecule that binds to the enzyme outside of the active site, changes the shape of the enzyme. So the enzyme undergoes some sort of conformational change that affects the activity of the enzyme. It can either activate the enzyme, it can inhibit the enzyme, but it's not binding in the active site, it's binding somewhere else, and it's altering the activity of the enzyme. You can also have covalent regulation where you're putting some sort of post-translational modification, such as a phosphorylation on the protein, on that enzyme. And that is also can cause a conformational change in the enzyme. It can also open up a binding site for some sort of partner that maybe it helps activate or inhibit it, that sort of thing. And so that would be covalent regulation as opposed to when you have these small molecules binding that you get this allosteric regulation. Now, often those allosteric regulators are going to be intermediates of either the same pathway or of a different pathway. So if one of these regulators that's listed is in the same pathway, then we have the options of it probably either being feedback inhibition or feed-forward stimulation. And so feedback inhibition is where you have a downstream intermediate that inhibits an upstream enzyme to ensure that supply doesn't exceed demand. So like stop sending me stuff. Whereas feed-forward stimulation would be where you have an upstream intermediate that's activating a downstream enzyme in order to ensure that supply meets demand. So get ready because here substrates come. So if you think of an assembly line, you have pieces that are coming down the pathway, and so you have a series of enzymes that are helping piece them together. The enzymes at the end of the pathway, those workers, they don't really need to be active and doing stuff if there's nothing coming their way. But once there is stuff coming their way, then you want to wake them up and have them get ready to work. But then they go to work and then they make lots and lots and lots, but they're making so much and there's not being used. And so it's like, why are we working so hard making so much of this if no one's using it? Stop sending us these pieces to put together. And so that would be feedback inhibition as opposed to feed-forward stimulation where we're waking up the workers. Now we want these workers to kind of go to the workers at the very start of the pathway and get them to stop doing their work. So in a more metabolically relevant context, if we look at glycolysis, we see examples of both feedback inhibition and feed-forward stimulation. So hexokinase is inhibited by its product, glucose 6-phosphate. So hexokinase 4, the version in our liver and our pancreas, is not inhibited by G6P, which allows them to then serve as glucose sensors and to take in excess glucose if needed. They aren't inhibited by G6P, but the other isoforms, the other versions are. And this is an example of feedback inhibition. And it makes sense because you don't want those tissues that have those higher affinity transporters and stuff, you don't want them to be taking in all the glucose when it comes their way if they can't actually use everything that they're taking in and trapping. Especially because most tissues don't have glucose 6-phosphatase, so they can't kind of untrap the glucose once they phosphorylate it. And so this feedback inhibition is important to keep these tissues from taking in more glucose than they can use, and therefore stealing the glucose, hogging the glucose from the rest of the body, and making blood sugar dangerously low. An example of feed-forward stimulation, if we look at pyruvate kinase, one of the things that it's activated by is this F16BP. F16BP is fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate was an intermediate that was produced upstream in the pathway. And so this fructose 1,6-bisphosphate that's made up here is now going to bind to pyruvate kinase and cause pyruvate kinase to become more active. And this is then going to allow it to kind of help drive this pathway forward, get the pathway ready in order to meet the supply that's coming its way. You also want to look to see if that regulator is generated in a different pathway. And so sometimes it can be generated in multiple pathways, or it can be generated in a different pathway and influencing this pathway. An example of this we can find in our fatty acid metabolism. And if we think about fatty acid metabolism, when we're making fatty acids, that's going to be happening in the cytoplasm. And when you're breaking down the fatty acids, that's in the mitochondria. During the synthesis, you have this key regulatory point, this acetyl-CoA carboxylase, this ACC. It activates the acetyl-CoA for incorporation by adding a carbon group to make malonyl-CoA. The breakdown, the key regulatory point of the breakdown is going to be the CPT1 or this CAT1, which makes this so that the fatty acid can actually enter the mitochondria to undergo the beta oxidation and get broken down. What happens is that the malonyl-CoA that's generated during the process of making fatty acids can bind to and inhibit that CPT1 or that CAT1 to inhibit the breakdown of the fatty acids so that you're not breaking down the fatty acids that you're making. So this is going to be an example of pathway crosstalk. In our fatty acid metabolism, we can also see examples of feed-forward stimulation and feedback inhibition. In terms of feed-forward stimulation, we see that citrate is activating this ACC. Now, citrate can be removed from the citric acid cycle, taken into the cytoplasm, converted back to acetyl-CoA, and used to make fatty acids. And so note that we can't take acetyl-CoA out of the mitochondria, but we can take citrate out of the mitochondria. So citrate we can generate from the TCA. We take it out of the mitochondria, we convert it back to acetyl-CoA in the cytoplasm, and then we can use it to make these fatty acids. And so citrate is going to go, and if we're building up a lot of citrate, maybe we can't use it all for the citric acid cycle. We want to instead use it to store energy as fat. What can happen is that the citrate can then go and bind to and activate this ACC and help stimulate the production of these fatty acids. When you're making those fatty acids, you're generating this malonyl-CoA, and this malonyl-CoA is now going to inhibit the breakdown of fatty acids. So this is an example of reciprocal or inverse regulation, where the same regulator is going to activate one pathway and inhibit an opposing pathway. We also see feedback inhibition, where kind of the end product of this fatty acid metabolism, you get this palmitoyl-CoA, so this 16-carbon fatty acid with the CoA attached, and that actually binds to and inhibits the ACC. So if you're making too much fat, you want to slow down that process. You want to inhibit the ACC. Other examples of inverse regulation can be found with things like NADH and NAD+, as well as ADP, AMP, and ATP. So ATP is going to allosterically inhibit glycolysis in the citric acid cycle. So stop making more ATP. We've got plenty. Let's instead go and use this ATP that we're making in order to help us make other useful things. And if we're using a lot of ATP, what we're left with is ADP and AMP. And those are going to allosterically activate glycolysis in the CCA so that we can then start running the pathway. We also see that NADH is going to inhibit catabolism. So remember NADH, it'll build up if you are kind of doing too much of those catabolic pathways and your electron transport chain can't keep up, or your electron transport chain is kind of clogged up for a different reason. There's no oxygen, something like this. The NADH is going to build up, and then it's going to inhibit various catabolic enzymes. We can also see more examples of feedback inhibition. We got our glucose 6-phosphate going back and inhibiting the hexokinase. We see oxaloacetate can inhibit this citrate synthase. So various examples of feedback inhibition that we're seeing here as well. But note that we have these same regulators that are regulating multiple different enzymes. So we can kind of have this coordinated regulation. Think about why that's making sense. And keep track of these regulators. So you have the regulators in the pathways tab organized by enzyme, as well as in the regulators tab organized by the regulators. So what regulates this enzyme? And then what does that regulator of this enzyme also regulate? And this is going to help you draw connections as well as see how a change in the concentration of one of these things can alter all of these different pathways and all these different enzymes. Think about what those enzymes have in common, what they have in different, and that sort of thing. So those are what for allosteric regulators, but we also have hormonal regulation. And so hormonal regulation is often going to be acting through covalent regulation, so where you're actually making covalent changes to an enzyme, and or via transcriptional regulation. So the Transcriptional regulation is altering the amount of the protein that gets made by altering the amount of the transcription of the gene. So make more messenger RNA of it to make more of the protein, or make less of the RNA of it to make less of the protein. So it acts on a slower time scale, but it's more efficient than just altering the enzyme that's already present. And so what you could have is you have signaling cascades that are then going to lead to the, say, activation of transcription factors to alter transcription, or that are going to lead to activation of kinases or phosphatases in order to actually go and alter the enzymes in a covalent manner, post-translational modification. So often phosphorylation, dephosphorylation. So for example, insulin and glucagon, they often have these inverse roles where insulin activates, say, a phosphatase, and then glucagon activates protein kinase A, and then the protein kinase A puts on the phosphate groups that are altering the activity of enzymes, and then insulin comes in and activates the protein phosphatase that then removes the phosphate groups, kind of reversing the actions. And so you can have these inverse action of hormones. And these hormones are sometimes listed on these charts, but when you see one of these hormones, you need to keep in mind that that's going to be acting indirectly. The hormone itself isn't getting into the cells, and so it's influencing things going on in the cells through some sort of signaling pathway. And these signaling pathways can often be complex and things, but often just by knowing the hormone, you're then able to kind of figure out a lot even without knowing the exact pathway that the hormone is kind of activating. So keep things to keep in mind. Identify your destination, identify your starting point. Find the set of arrows that connects them. Keep an eye on where you see arrows that are reversible, where you see arrows that are one direction, where you see kind of multiple arrows that indicating that things are multi-step. You want to look for key regulatory points. So look at those points where you have a lot of these regulators listed. Those are often going to be at branch points, at starts of pathways, at ends of pathways, these sorts of things. Look at the arrows and see what is going to be your substrate, what's your product, what's your kind of co-substrates, cofactors, what's coming and going, and keep those in mind. Especially keep in mind where you're generating the NADH, the FADH2, where you're using them, or the NADPH. Look and see where you're losing carbon dioxide. So where are you actually losing carbons? Keep in mind at points where you're actually splitting a molecule in two. So how many carbons do you have here? You have 6 carbons. And then how many carbons do you have here? You have 3 carbons. And so you need to think about, okay, well, everything here is going to be happening twice per everything that was happening up here. Where are you going to be using ATP? Where are you generating ATP? Keep track of these. Keep track of what different regulators of these different enzymes. Think about where they're happening in terms of what tissues, where inside of those tissues, inside of the cells. Characterize the types of regulations. Look for those interpathway crosstalk. Look for the examples of your feed-forward stimulation, your feedback inhibition, your reciprocal regulation, and think really critically about what you're seeing. Why does it make sense? And so hopefully this will help you see that these pathways really don't have to be scary. They're very logical, and kind of just taking them step by step, but also keeping in mind the bigger context can be really helpful. Speaking of really helpful, I hope this helped.